Okay. So now we discuss about types of neuroglia cells or glia. Basically, types of glia cells. Okay. So this one, two, three, four, five. Actually, it's a four because this is a subtypes: fibrous astrocytes and protoplasmic astrocytes. This is a one cells. So there is a basically four types of glia cells present in central nervous systems. So here this is the four. One. This is also one. 2, 3 and 4. This is a glia of central nervous systems. But in some authors there is one more glia cells, that is a radial glia cells. It is also given in a types of glia in central nervous systems. But basically radial glia cells is a precursor cells of astrocytes. It is a precursor cells. So from radial glia cells, astrocytes and oligodendrocytes are differentiated. So it is during a embryonic life or newborn baby, but in a mature persons, radial glia cells are very few in number. So we not considered here, but it is a given. Radial glia cells is also part of types of glia cells in central nervous system. But in some authors are only given three: astrocyte, microglia and oligodendrocytes. Ependymal cells not given. So but it's not a true. There is a if we give a mature classifications, so there is a four types of glia cells present in central nervous system. That is a astrocytes, microglia, ependymal cells and oligodendrocytes. These four types of glia cells present in CNS, central nervous system, while in a PNS there is only two types of cells. We talk about the PNS, peripheral nervous systems. There is a basically two types of neuroglia cells. Most important is a Schwann cell and second one is a satellite cells. But recently one more glia cells added in a PNS, means peripheral nervous systems, that is a enteric glial cells. So though enteric glial cells or enteric nervous systems are nowadays they are totally different. So whenever we classify the nervous systems, there is a central nervous system, peripheral nervous systems and enteric. So this is the very recent study. But we know there is no more studies available or no documentations available. So we only consider there is a two cells, Schwann cells and satellite cells in PNS. So now talk about the some structures of neuroglia cells in CNS. So there is a astrocytes, oligodendrocytes, microglia and ependymal cells. So most abundantly present in a CNS, it's a astrocytes. It's a most abundantly 40 to 60%. Numbers of are most abundantly present is a astrocytes. Astrocytes, astro means star. Astro means it's a star shape because it's look like a star. You can see here the star, starfish or star. Basically astrocytes is a two types, fibrous and protoplasmic. What is different between fibrous and protoplasmic? Protoplasmic is a very much branching. They have a very much or they are very much divided. But these branchings are small in size. So numbers of branching is more, but size is a less. But in a fibrous there is a numbers is a less of branching, but it's a very sharp. You can see there is identical, but it's a long. While in a protoplasmic it is a short, but number is a more. It's reverse. Number is a less, but size is a very much long. So this is a fibrous and protoplasmic. Generally, fibrous astrocytes remain in a white matter, and protoplasmic remain in a gray matter. So what is white matter? White matter, you know, there is white matter is a bundle or collections of axons, or so you can say it's a collections of fibers. So now you know there is a fibers. So fibers is a collections of or there is a long axons, it's ultimately called as a fibers. So fibers and fibrous. So now you can memorize. Fibrous are remain in a fiber. Fiber is a white matter, and protoplasmic remain in a gray matter. This is only for hint how you can memorize, because now you can easily memorize, but after collections of topics, after if you want after the physiology, all physiology, that time you forget where is a protoplasmic and where is a fibrous. So fibrous in a fiber, what is fiber? Fiber is a white matter actually. Fiber means a collections of axons. Okay, then talk about the oligodendrocytes. Oligo means very few. Oligo means few. Dendro means tree like branching. Oligodendrocytes are very few branching. Okay. But its a size is a smaller than the astrocytes. It's a smaller, and number of oligodendrocytes is a less from astrocytes, but they are second in number. They are remaining 20 to 30% in CNS. Number is a 20 to 30%. And main functions of oligodendrocytes are they are responsible for myelination in CNS. It is a main function. So we also describe the functions here. But astrocytes we describe later, because it's a very big topic. It's a very full. We require the full understanding of astrocytes. But oligodendrocytes only responsible for myelinations in a central nervous systems. What is myelinations? Myelinations means covering around the axons. These are responsible for, they give a fast velocity, increase the velocity of action potential, plus they also give insulations in a central nervous system. So this is the oligodendrocytes. One oligodendrocytes give a to multiple axons. They give a multiple myelinations to axons. A single oligodendrocytes, you can see here there is only one oligodendrocytes, but they give a myelinations around the axons in a two neurons, two axons. While in a PNS, a Schwann cells responsible for myelinations, but one Schwann cells give a one myelinations. They are not occupied the multiple neurons, multiple axons in PNS. While one oligodendrocytes give to many more, one then more axons, myelinations. They cover the more axons. Then microglia. So microglia, it's a very smaller, it's a some branching, but it's a very sharp branching. It's a small, very sharp spine-like projections from a cell body. A microglia is originally, this is not originally from CNS. Microglia is a derivative of monocytes. During we discussed about the derivative of monocytes in a previous lecture, that monocytes are responsible for your immunity response. So some monocytes remain in some organs for permanent, like in a liver there is a Kupffer cells. So this microglia is also a derivative of monocytes, but this is a first differentiated microcytes from early life, from and during a developing life. So this is the earliest micro or monocytes who differentiated in microglia. This microglia can movable in CNS. They can move from one place to another place, but they cannot come out from CNS. Microglia are responsible for phagocytosis, means they give immunity from foreign material, foreign particles, bacteria, virus, fungi, parasites, whatever. So they responsible for phagocytosis, means they engulf the foreign body and they destroy that. Also they also responsible for clean out the some waste material from CNS or debris of CNS. They cleaning out the waste material which is origin after some injury. So microglia are responsible for mainly for immune reactions phagocytosis. So they give a protection to CNS from foreign body or foreign particles. Now next is ependymal cells. What is ependymal cell? Ependymal cells is generally of columnar cells. You can see there is a cuboidal or columnar cells. They are mainly remain in a ventricle systems of brain and central canal of spinal cord. You know, there is a in our brain there is a multiple ventricle system is there. Lateral ventricle, third ventricle, fourth ventricle. It is a hollow space which is occupied by a special fluid. It's called as a CSF. So this flow, these ventricles are suppose this is a ventricle. So the ependymal cells form a single layer network in a floor of ventricle, and they have a microvilli, cilia, villi and microvilli is there. They are moving. So ependymal cells are responsible for formations of CSF, directions of CSF, and they also maintain the internal environment. So they maintain the constant environment of CSFs. So they also are responsible for maintain the ionic balance or compositions of CSF, cerebrospinal fluid, which is our next topic. So ependymal cells, it is a columnar cells, remain in a ventricle in a single layer. They are responsible for synthesis or flow or directions of CSFs, and they regulate the CSF environment or regulate the components of CSF. So this is about the neuroglia cells in a CNS. Now neuroglia cells in a PNS. First one is a Schwann cell and second is a satellite cells. Schwann cells are responsible for myelinations around the neuron or axons in a PNS, not in a CNS. CNS are done by oligodendrocytes. So myelinations in CNS is a different and PNS is also different. But Schwann cells, they only make one Schwann cells make only one neuron. They not participate in multiple neuron. So whatever axons are myelinated with Schwann cells or oligodendrocytes is called as a myelinated neuron. Or some neurons are not having a myelinations. This is called as an unmyelinated neuron. Okay. So how I will explain how this myelinations occur. You can see here they wrapping around the neurons, multiple fold. They produce the multiple myelins protein. It is a fat and protein materials. So this fat and protein material around the neurons in a PNS is a different, and myelinations or myelin protein or fat is around the neuron in CNS is a difference. Why it's a difference? So it is important. Some disease affect the myelin protein and fat. So whatever disease affect myelin proteins, if affect only CNS, they cannot affect in PNS because both protein and fat material are different. So suppose there is an autoimmune disease called as a Guillain-Barré syndrome. They affect the myelin of PNS only or Schwann cells myelin. They cannot affect the central nervous system. Why? Because this myelin are different. They formed by the oligodendrocyte is some different, but their functions are same. Myelinations. They give insulations. They give increase the velocity. It's called as a saltatory conduction. So I will give examples. Suppose this is a axons. So they produce the myelin sheath around the neurons in a multiple fold times like this. So axons are covered by multiple fold. You can see here there is a multiple fold, and most important there is a nuclear part are remain on surface. So this is the axons. You know axons having exoplasms. So the axons are covered. They are covered by a exoplasmic membrane. This is called as exolemma. This axons covered with the multiple fold of myelin sheath. This is called as a neurolemma. Okay, because axons having a two membrane. One inner membrane is a exoplasmic membrane, and they are surrounded by a like this myelinations, multiple fold of Schwann cells. So this is about the Schwann cells. Now satellite cells, there is very few informations available for satellite cells. It is also flat cells, and they remain in a ganglion of PNS, and they are responsible for give us support and nutrition to a cell body of PNS, which is remain in a peripheral nervous system, ganglion of peripheral nervous system. So this is all about the types of neuroglia cells. We talk about the fibrous astrocyte, protoplasmic astrocytes. We not still discuss about the functions of astrocyte, but we discuss the functions of different microglia responsible for immunity, means they are responsible for phagocytic activity. Ependymal cells, which are responsible for formations of CSF, directions of CSF, as well as they maintain the environment of CSF or content of CSF. Oligodendrocytes, they are responsible for myelinations in a central nervous system. Next, in a PNS, Schwann cells, they give a myelinations in a peripheral nervous system, and satellite cells give us support and nutrition to cell body of peripheral nervous systems. That is a cell body present in a ganglion of peripheral nervous system. So now, further we talk about the astrocytes, which is most important. Previous year, a scientist thought that neuroglia cannot generate the action potential. They cannot transmit the action potential or electrical activity. But recently, that shows astrocytes having some electrical activity. They can also transmit the current. So now, after break, we talk about the astrocyte in a detail.